While on the surface, the coral looks like a simple, humble animal, indeed it is a microcosm of diverse life. Many critters call the coral polyp home. Dubbed the coral holobiont, the main reef-building organism is a source of bacterial, archaeal, eukaryotic, and viral diversity. Together, these organisms are able to live in harmony. For example, the coral live in a happy symbiosis with the single-celled zooxanthellae. These photosynthetic algae reside in the tissues of the coral and produce oxygen, sugars, and other nutrients the coral and its microbial inhabitants need to survive. However, recent research has raised the question, is this such a happy symbiosis? A more recent hypothesis is that the coral actually farmed the zooxanthellae, incarcerating the helpless cells to benefit from the antiparasitic armor, an easy source of nutrients. However, stressors, often induced by human intervention, such as temperature fluxes, changes in pH, and nutrient loading, threaten the fragile ecosystem. The coral lose their tight grip on the zooxanthellae as they are expelled from the coral tissue, resulting in an event known as coral bleaching, a dramatic loss of color provided by the algae. Although these coral are not dead, without their armor they are left crippled, susceptible to many diseases. These diseases include black band syndrome, skeletal eroding band, white syndrome, brown band disease, growth anomalies, tissue necrosis, and cyanobacterial syndromes. For many of these diseases, the opportunistic pathogen has been identified using Koch's postulates. Coral disease affects coral reefs all over the world, in areas such as the Great Barrier Reef in Australia, with a high majority of diseases reported in the Caribbean as well. Scientists predict that by 2050, most of the reefs will be destroyed at the current rate of decline.
The abundance of microbial species in reef ecosystems leads to the bacterial challenge. Diseases become prevalent in coral ecosystems after a bleaching event, making it even more difficult for coral to recover. Is bacterial bleaching more common than once thought? Research is telling us yes. Bacterial populations in coral often change in response to bleaching events, and some bacteria that aid in recovery from ecosystem stressors are often outcompeted by bacteria that thrive during high temperature and induce bleaching. The increase in ocean temperature is also thought to make some bacteria more virulent, which causes the enhanced bleaching episodes. With consistent and increasing bleaching events, research into the effects of bacteria and high temperatures on reefs is a growing branch of marine ecology. Common reef coral M. digitata was treated with 5 pathogenic bacterial strains that are commonly found around the coral holobiont at high temperatures. Bleaching occurred, resulting in discoloration and reduced zooxanthellae density. The damage of bacteria coupled with coral bleaching has visible effects on the coral branch structure. At 27 degrees Celsius, without the addition of bacteria, no bleaching was observed and the coral was determined to be healthy. At 27 degrees Celsius, with the addition of 5 mixed bacteria, minor bleaching was observed and the coral was still determined healthy. At 32 degrees Celsius, without the addition of bacteria, minor bleaching was occurred and the coral was determined somewhat compromised. At 32 degrees Celsius, with the addition of all 5 mixed bacteria, intense bleaching and brittleness occurred. At 32 degrees Celsius, with the addition of just the Sulfidobacter species, the bacteria that was determined to be the most indicative of coral bleaching, intense bleaching and tissue necrosis was observed. At normal temperatures, bacteria did not cause coral bleaching, affect metabolism, or antioxidant enzyme activity in M. digitata. However, at high temperature stress, addition of bacterial strains caused a decrease in zooxanthellae density. Zooxanthellae density decreased by 70% compared to the control. When the symbionts leave the coral, the host doesn't receive the nutrients the symbiont was providing, making the coral weak and even more susceptible to bacterial disease. The same negative effects was observed in photosynthetic efficiency, decreasing by 25%, and photosynthesis and primary production showed a large decrease at high temperatures coupled with bacteria. Zooxanthellae are photosynthetic and involved in providing the coral with essential nutrients. Decrease in primary production will affect the whole ecosystem. Results showed a major primary production decrease even after just being exposed to the bacteria for 4 hours, showing that bacteria affects coral ecosystem processes quickly. Calcification rate was most affected by high temperature and bacterial combination. decreasing by 101% by day 8. Because calcification is very energetically costly, this dramatic reduction could be correlated with decrease in zooxanthellae, because that causes the decrease in energy. This impacts calcium carbonate deposits, reduces coral skeletal strength, and is likely inhibitive of new coral skeletons from forming new coral reefs.
Coral reef biologists are working on detecting disease outbreaks. Functional genes may be the key to understanding this complex system of biotic and abiotic interactions. Scientists can measure the genetic diversity of healthy and diseased corals to understand the interactions within the holobiont. Microbial symbiosis is marked by the loss of functional gene diversity, suggesting that tightly symbiotic bacteria lose genes they no longer need, such as the membrane transport and cell signaling pathways. This tight symbiosis may be lost in diseased corals as they show a greater gene diversity. Genes key to biogeochemical cycling of carbon, nitrogen, and sulfur vary between diseased and healthy coral. For example, all the genes necessary in the complete nitrogen cycle are found within the holobiont. However, in diseased corals, ammonia detoxifying genes are more diverse, suggesting that ammonia waste is higher in diseased corals. This also means that bacteria capable of ammonia oxidation may be critical to detoxifying ammonia in healthy coral. Sulfate is also a disease indicator, as coral with black band disease is caused by sulfate-reducing and sulfur-oxidizing bacteria. The number of DSR genes, the genes involved in sulfate reduction, increase in diseased corals. Finally, it comes as no surprise that in unhealthy coral, the number of virulent genes increase, the genes such as genes that allow the pathogen to be motile and attack the host and secrete toxic compounds.
Many wonder if the coral can adapt fast enough to develop resistance to disease and environmental stress-related conditions. The coral probiotic hypothesis proposes that the coral holobiont is a collection of the most adapted microbial inhabitants. This hypothesis suggests that the diverse bacterial population associated with the mucus and tissue in this community undergoes a rapid change when environmental conditions are altered. This helps explain the coral development of resistance to pathogens, even though the coral lack an adaptive immune system. Thus, the bacteria in the holobiont is similar to humans eating yogurt to promote a healthy microbiome. The question that arises is, can coral microbial communities recover after bleaching events? One study of the microbial populations of a hard coral, Acropora millipora, showed complete recovery of coral zooxanthellae densities to pre-bleaching levels after a 1-month bleaching period of increased ocean water temperatures. Other microbial populations, unfortunately, are not so lucky. As long as the carbon pool of the atmosphere continues to increase, ocean acidification and coral bleaching will continue to be a problem for coral reef ecosystems for years to come.